Hello everyone, welcome to GChem classes. So today I am going to discuss very hot topics, and it is very easy topics, and I think 1 hour or 2 hours if you are giving, your entire bioinorganic will get covered. Okay, I am discussing bioinorganic today. So entire bioinorganic will get covered, and 1 or 2 questions surely they are going to ask from bioinorganic, and it will be very much easy. Let's see. Together we will discuss it. First we will see the concepts, and then we will directly go to the PYQs and we will solve it, correct? So let me start. Bioinorganic. Okay, so in bioinorganic we have those elements, right? We know that elements is required for body functioning, but there are many elements which is not required. So likewise we will classify our elements in two different classes. One will be our essential elements, and next will be our trace elements. We have many more like non-essential elements, toxic elements, that we are not going to discuss it, correct? So in your syllabus IIT JAM 2023, you just see it has clearly mentioned essential and trace elements. So now just we will see essential elements and trace elements. Okay, so essential elements means it is very much required for your body function. The main requirement element is carbon, hydrogen, oxygen, sulfur, nitrogen, magnesium. Okay, because we know that in our body, okay, whatever we have made up, it is a carbon, hydrogen, oxygen, right? Glucose, carbohydrate, correct? Protein, correct? We are taking the oxygen, dioxygen, correct? So everything is like we can categorize it like carbon, hydrogen, oxygen, nitrogen. These are the major elements which we need. Approximately 98% our body is made up of carbon, right? Then nitrogen, we have sulfur. Then what else we have? We have magnesium. Okay, we have magnesium. So likewise, these are the essential elements. In trace elements, we require trace elements is required like in our body for like smoothly this digestion process or any other cycles like Krebs cycles. Okay, for this functioning we need enzymes, and enzymes, the coenzyme part, in that coenzyme part the active site it will present a metal, right? So that those metals we require like iron for hemoglobin, correct? Then we have copper, we have zinc. Okay, these are the trace elements which we need, correct, for our body functioning. So there are many, so etc. Correct. I think 5 or 6 heavy metals are there which is required for body functioning. Okay, so iron, copper, zinc. Okay, we need manganese, we need cobalt, we need iodine. Okay, so these are the traces elements which we need. Now this is the classification based on essential and trace elements. We have one more type of classification that will be based on your metal, like which heavy metal is added up there. Based on that we will just classify. So we'll just write here bioinorganic. Okay, and then we'll classify. One will be like it's required more places. Okay, so first will be oxygen storage based on function, right? Oxygen storage and transport, correct? Next will be, first will be oxygen storage and transport. What we will do it, just take it longer so we can fill it up easily in this place. Okay, next is metalloenzyme, nitrogen fixation, okay, and then metal storage and transport. Okay, metal storage and transport, and then next will be photosynthesis, okay, and then nitrogen fixation is done, metal storage and transport, photosynthesis, and then next will be electron transfer system. So here we'll just classify. Here oxygen storage and transport, we will have heme and non-heme. Okay, for heme we require iron, for non-heme we require copper. Okay, we'll see those examples in detail. Okay, iron, we have hemoglobin, correct, for transport, and for storage we have myoglobin. Okay, in these are for like human beings and whatever the vertebrates are there. Okay, for vertebrates, and this non-heme is required for your invertebrates. Okay, invertebrates we will have copper. Even we have iron also, hemerythrin and hemocyanin, right? Hemerythrin will have iron, hemocyanin will have copper. So we'll discuss in detail. Then metalloenzyme, carbonic anhydrase, okay, carboxypeptidase. So this we will discuss here. Nitrogen fixation. So nitrogen fixation we need molybdenum, right, for nitrogen fixation, that we will see it. Then metal storage and transport. So metal storage and transport. In metal storage you see the ferritin, transferritin, right? Then photosynthesis. Photosynthesis, we know that magnesium is very much requirement because the chlorophyll center, okay, it is having the magnesium, correct. And then electron transport system, reduction, okay, those we will see. Okay, iron protein, that will help you out here, right? In this bioinorganic, the next subtopic, we can say the subtopic. So it will be the how many types of rings it is involved in bioinorganic, that we will check it out, right? So first will be your porphyrin. So rings, I just write rings. Okay, it is very much important because so many times they have asked that for vitamin B12 which ring it is. Okay, the ligand ring. For chlorophyll which ring it is, and for your vitamin B12 chlorophyll, and then we have hemoglobin. So for hemoglobin which ring it is functioning, correct. So just we'll discuss three types of, there are many types, but three types of rings we are going to discuss, which is important for you. One will be porphyrin. Okay, porphyrin ring will be like, you have to just draw it. Okay, and then okay, this is CH2, 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 correct. So we are drawing porphyrin ring, correct. So porphyrin ring, it will be like okay, 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 okay. How many other? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So 11 double bond. Okay, so porphyrin will have 11 double bond. Here it will be hydrogen, here it will be hydrogen. This is porphyrin ring, correct. So if I put metal here, then it will be metalloporphyrin, right? So suppose if I am putting iron here, then it will be myoglobin, right? It will be attached to histidine, and then this side it will be O2 or H2O, okay, whatever is there. So this porphyrin ring, we can say the example where it will be found. It will be in hemoglobin, okay, myoglobin. Okay, next we will take for vitamin B12, right? That we call choline ring. Okay, let me draw that one here itself. Okay. Okay. CH2, CH2, CH2. Here one CH2 is missing, correct. Okay, so now we'll see the double bond, how it will be. 1, then 2, then 3, 4, 5, and then no, it won't be here. Okay, so it will be 1, 2, then 3, then 4, then it will be 5, and here 1 will be 6. So 1, 2, 3, 4, 5, 6. So there will be 6 double bond in choline ring. Okay, so choline ring example will be coenzyme. Okay, coenzyme B12. So vitamin B12 we have. So coenzyme B12. Okay, so suppose here one metal is there. So vitamin B12 we know that it will be cobalt. So if cobalt is there, you just see they might be asking how many 5-membered rings are there and how many 6-member rings are there in this porphyrin ring. So how many 6 members are there? 1, 2, 3. So 3 6-membered rings. How? See, 1, 2, 3, 4, 5, 6. Here also 1, 2, 3, 4, 5, 6. Here also 1, 2, 3, 4, 5, 6. So total 3 6-membered ring. And how many 5-membered rings are there? 1, 2, 3, 4, and this one is 5, right? See, 1, 2, 3, 4, 5. So total 5 5-membered ring. Okay. Likewise, here also they might asking you for this. Suppose I am putting iron here. For hemoglobin, we'll put iron. So how many 5-membered rings are there and how many 6-membered rings are there? So 5-membered rings are there. See, 1, 2, 3, 4. So 4 5-membered rings. And how many 6-membered rings are there? 1, 2, 3, 4. So 4 6-membered rings. So in porphyrin ring, 4 5-membered rings and 4 6-membered rings will be present. Okay, just count 1, 2, 3, 4, 5, 6. Right? You can count any of these rings. It will be 6-membered itself, right? 6-membered only. So now porphyrin is done, chlorine is done. We just draw for chlorine ring that is required for chlorophyll, right? So I will utilize this ring. I will utilize this ring and then we'll see. I'll just remove this porphyrin. I'll put chlorine. Okay, chlorine ring. Example will be chlorophyll. Okay, what will happen? One double bond will be missing. That's all. There will be total 10 double bond. Okay. So porphyrin ring 11 double bond, chlorine ring 6 double bond, and your chlorine ring will having 10 double bond. Okay. So this is a chlorophyll. So here also, suppose we are putting magnesium here. How many 6-membered are there? 4 ring are there for 6-membered. How many 5-membered are there? So 4 5-membered rings. So these might be they asking in NAT type question. Okay, numerical aptitude type question. They might be asking. Correct? One will see. First we will see it for hemoglobin, and after hemoglobin we'll see it for myoglobin. Okay. So for hemoglobin, I'll just draw the structure, and then it will be very much easy to explain the things. Okay. Okay. Whenever you are drawing this type of ring, porphyrin ring, first draw this body structure and then try to put double bond and all. Okay. So it will be like easy for you. Then here, then it will be here, then it will be here, 1, and then here, then it will be here. So how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So 11 is done. So this is your we are making hemoglobin unit. So here it will be histidine, and this side it will be OH2. So likewise, we have one more porphyrin ring. We have one more porphyrin ring, and we have one more porphyrin ring. So total 1, 2, 3, 4. One hemoglobin unit will have 4 porphyrin ring, and whatever this bond you are seeing, this we call salt bridge. Okay. Each porphyrin is attached with the another porphyrin ring. In between we have salt bridge. Okay. By the help of salt bridge, it is attaching. Okay. Here also salt bridge. Here also salt bridge. Here also salt bridge. So total 4 salt bridges are there for 4 porphyrin ring, and all together we call it it's a hemoglobin. You see that oxygen dioxygen is to iron ratio, it will be 1:1, right? For 1 iron, 1 oxygen will be there. So this is deoxyhemoglobin. If we are just removing this OH2 and just we are putting O2, then it will be oxyhemoglobin, correct? So we have two types, deoxyhemoglobin and oxyhemoglobin. So now for this oxyhemoglobin, if you will see the ratio for iron and oxygen, it will be 1:1 for hemoglobin, right? And this will carry, this is all about hemoglobin. Now we'll discuss about myoglobin. Okay, this is for transportation. Okay, this hemoglobin is working for transportation oxygen. Now we'll see for myoglobin, which will be the storage for oxygen, right? Myoglobin, okay, and it is storing dioxygen in human, like the vertebrates tissue. Okay, tissue, you know, right? It's a constituents of cells. Okay, so in tissue it will just store the dioxygen. So we'll see that myoglobin structure first. So it will be again same, porphyrin ring will be there. I'm not okay, I'll draw it, no problem. Okay. So the many times you will draw, it will be more perfect, right? Okay, porphyrin ring we are making. Okay, these are the porphyrin ring we are making. Okay, then this double bond is here, here one double bond here. Okay, and then this one is here, this one will be here. So see, here also 4 5-membered ring and 4 6-membered ring for myoglobin also. Here also it is attached with histidine. Here it will be attached with O2, so this will be oxymyoglobin. And if it is attached with OH2, then it will be deoxymyoglobin. Now for hemoglobin, the difference between hemoglobin and myoglobin is, for hemoglobin we need 4 unit. Okay, and there will be salt, and there it is there it is attached by salt bridge, correct? For myoglobin only single unit itself is enough for myoglobin, correct? Next we'll see hemerythrin. Okay, we'll see the hemerythrin, how the structure is there, because see this structure you no need to remember it. Okay, just look 1 or 2 times, because there in examination you will have 4 options, correct? So based on that you'll just easily identify. Suppose they are asking for hemoglobin, which is the central metal, so iron. For myoglobin, which is the central metal, iron. For what is the peripheral ring it is attached, like ligand ring it is attached, you can easily say it's a porphyrin ring, correct? How many double bonds are there? So 11 double bonds. Anything they might ask, okay, basics question. So just have a look for structures and see their function. Okay, structures and function, and mainly the metal. Okay, what metals is present in that. Okay, so hemerythrin, we'll see the hemerythrin. Okay, okay, hemerythrin will contain it is a like storage and transportation. Okay, storage and transportation of dioxygen in invertebrates. Okay, invertebrates, they store and transport for oxygen, because both the functions together it is doing this hemerythrin itself, correct? So we'll have iron, and here also we have iron, correct? Okay, then it will be attached with aspartate, then it is attached with glutamate. Okay, here 2 histidine unit will be there, here 3 histidine unit will be there. Okay, so this is your deoxyhemerythrin. Okay, if you want oxyhemerythrin, it will be oxygen and then oxygen. Okay, here it will be hydrogen. So this one, so this will be oxyhemerythrin. Okay, this one will be oxyhemerythrin, and if you're removing this O2, then this will be deoxyhemerythrin, and they might be asking you what is the metal center. It will be iron and iron, and they might be asking what is the coordination number of each metal center. So one iron will have 1 2 3 4 5 6, so this will be hexa coordinated. Okay, hexa coordinated, and this one 1 2 3 4 5, this will be penta coordinated. Okay, this will be penta coordinated. So maybe they might be asking these questions, otherwise no need to remember, just if you know the structures easily you can calculate it. And this penta coordinated will get changed when it will be oxyform. Okay, if oxygen is attaching, then this will be oxyhemerythrin, correct? So once it is forming oxyhemerythrin, then what will happen? This will be penta to, this is going to change for hexa coordinated, correct? So that you have to remember whether they are asking for deoxyhemerythrin or they are asking oxyhemerythrin. If simply they are asking for hemerythrin, the number of coordinated bond, so you have to see hexa and penta. See the hemocyanin, okay? Let me see the hemocyanin. Okay, metal center is very much required, है ना? Hemocyanin. This is also storage and transportation in invertebrates. Okay, invertebrates. Okay, here also it is for invertebrates, because for vertebrates we know that we have myoglobin and hemoglobin, correct? So this hemocyanin, how it will be? First of all, this is written heme, okay, but it is a non. What we see in this hemocyanin, heme it is written, but iron is absent here, right? First time you are seeing. Okay, so here it will be copper, correct? So they might be asking this question, copper and copper. So what we will do it? Let's take histidine, 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 and there will be hydrogen bond, correct? In between histidine and histidine. So this will be deoxy form, correct? This will be deoxy form of hemocyanin, and hemocyanin we call it blue blood. Okay, due to copper, okay, it is called blue blood. Okay, this color appearance is blue color, right? So blue blood. So now you see this is deoxy form, okay, and if it is in oxy form, how it will be like oxygen? How it is going to attach the dioxygen? It will be one oxygen here, another oxygen will be here, correct? In between bond will be there. So now you see it will be in bridge form. This is oxyhemocyanin, correct? So just see, if you are removing these two, there will be no bond between copper and copper, okay, direct bond. So if you are removing these two, then it will be deoxyhemocyanin. If you are adding this one, then it will be oxyhemocyanin, correct? It is present in invertebrates. It's called blue blood. Okay, coordination number if they are asking, easily you can say it, correct? So this is hemocyanin. Now we'll see, directly we'll go to the enzymes, okay? Enzymes and their metal center, correct? And then we'll solve few PYQs, which is very much required. Otherwise, how we'll verify that whatever we are learning, whether it's useful or not? Metalloenzyme we'll see. Okay, in metalloenzyme, first we'll see carbonic anhydrase. Okay, carbonic anhydrase. What carbonic anhydrase will do? It will do your CO2, whatever carbon dioxide is there in presence of water, it will be changing to H2CO3. Okay, carbonic acid, it is changing to carbonic acid. For this we require carbonic anhydrase. So see the enzymes active site. Here it will be OH2, and this OH2 is reacting with this carbon dioxide, right? So here it will be histidine. Here also it will be histidine. Here also it will be histidine. So this will be your enzyme active site, active part, where the metal center will be zinc, correct? For this carboxy anhydrase, the metal center will be zinc, and the function is the whatever carbon dioxide is there, that they will change it to carbonic acid, correct? So just it is converting the carbon dioxide to carbonic acid, correct? So carbonic anhydrase. So second we'll have, here only we'll draw it, so both like together the enzyme will be there. Carboxy peptidase, okay? Carboxy peptidase. Okay, so what is the function of this carboxy peptidase? This will cleave that peptide bond. Okay, peptide bond we know, right, that two amino group will be joined together. Okay, so peptide bond, it will cleave the peptide bond, but we call it exopeptidase. Okay, what is exopeptidase, endopeptidase? Exopeptidase means from outer side it will, suppose this is one amino acid, these are the amino acids for this protein. If it is cleaving from ending, first it will cleave this one, and then second it will cleave this one. Okay, first, second, then third it will cleave this one, fourth it will cleave this one. If it is endopeptidase, what it will do? Directly it will go to in middle and it will cleave it. correct, for endopeptidase. And for exopeptidase, it will start from end side, okay, 1, then 2, then 3, then 4, okay. From end, if it is cutting, it is exopeptidase. In middle, if it is cutting, then it will be endopeptidase. So this enzyme, particular enzyme carboxypeptidase, is a exopeptidase, okay. So how it will look? It will be zinc OH2. Why I have written together? You just see the difference. Histidine, and here it will be glutamate. Correct? Glutamine. So this one is getting changed. One histidine is replaced with glutamine. Your carbonic anhydrase is changes to carboxypeptidase. Okay. So these are the enzymes. There are many enzymes. So better we'll go for PYQs and we'll solve it. There in PYQ we'll solve for vitamin B12 also. Okay. So let me see the PYQs. Okay. First question is from 2022. Okay. Question number 8. We'll see how we are going to solve it. You can see in this screen the question. Okay. Question, it is appeared in the screen. You can directly see. So first question. Among the following, the biomolecules with a direct metal carbon bond. Okay, metal carbon bond it is asking. Question number 8. Okay, in 2022. Question number 8. So metal carbon bond they are asking. First they have given coenzyme B12. So coenzyme B12 is vitamin B12. Which ring it will having? Corine ring. Okay, corine ring. Let me draw the corine ring. CH2, CH2, CH2. Here won't be any CH2. Okay, total 6 double bond will be there. Let's draw it. Okay, 2, 3, 4, 5, 6. Okay, 1, 2, 3, 4, 5, 6. 6 double bond is there. Center it will be cobalt. Okay, there are 5 member and 3 6 member rings are there. And then in this cobalt, down it will be added histidine. Here it will be X. Now this X is depend on, if it is cyano group, then we call it cyanocobalamine. Okay. If this X is CS3, then we will call it methanocobalamine. If this X is adenosyl, okay, if it is adenosyl, then we call it coenzyme B12. And this cyanocobalamine only we call it vitamin B12. But in your question they have asked coenzyme B, like biomolecules with a direct metal carbon bond is. So your cyanocobalamine will also have direct metal carbon bond. Okay. Your methanocobalamine will also have direct metal carbon bond because this X, okay, it is directly connecting. Suppose in this place, if we have CN, it will be metal carbon bond. If it is having CH3, then also metal carbon bond. If it is having adenosyl group, you know the adenosyl group, the 5th position is a carbon, right? In that 5th position carbon, it will attach for adenosyl, okay? So there also, it is a metal carbon bond. So I think your option A will be correct. Coenzyme B12, okay. Nitrogenase we will have molybdenum in center. Chlorophyll we already know that porphyrin ring. This is a corine ring. For chlorophyll it will have your chlorine ring, and in chlorine ring we know that for chlorophyll it is magnesium. Okay, center is magnesium. With nitrogen it is connecting, right? Metal is connecting with nitrogen, not with carbon. So there won't be any metal carbon bond in chlorophyll also. In hemoglobin we have porphyrin ring. We already discussed, right? Porphyrin ring. So in that porphyrin ring center will be iron. So there also what we have, iron, one side will be O2 and another side it will be histidine. So if it is connecting with histidine, then it is connecting with nitrogen, not metal carbon bond. Okay. Okay, question number 38. You can see in the screen. From the same year, okay, 2022, question number 38. What it says, zinc containing enzymes is, okay, zinc containing enzyme is carboxypeptidase, zinc contained, okay, and then hydrogenase, no, it is not contain zinc. Carbonic anhydrase, it contains zinc. Urease also, I guess urease is containing nickel, okay. So carboxypeptidase and carbonic anhydrase will be your answer, because we already saw, right? Carbonic anhydrase and carboxypeptidase. We know that this is a exopeptidase, and for carbonic anhydrase, carbon dioxide is converting to your carbonic acid. Okay, carbonic acid. So for that, this zinc, 3 histidine were there and 1 water. For carboxypeptidase, 2 histidine, 1 glutamine and water. Center was zinc. So your option answer will be A and C, correct? We'll see the next question. From this year, there won't be any other questions. Okay, we'll see the question number 6 from 2019. The question says the correct option for the metal ion present in the active site of myoglobin, hemocyanin and vitamin B12. Okay, so myoglobin, we know that it's iron. Okay, for myoglobin, we know that it is iron, and already we drawn hemocyanin. Okay, for hemocyanin, we know that it is copper, already we drawn. And for vitamin B12, okay, vitamin B12, it is cobalt. Let's see, iron, copper, cobalt, iron, copper, cobalt. Option C will be your correct answer, right? Now going to the question number 35. They'll ask very easy question from your bioinorganic chemistry. That's why I took this topic, and let me clear first. So we'll directly go for organic and physical, right? So this question will solve it. So question number 35, what this says, the true statement regarding the carbonic anhydrase enzyme. Carbonic anhydrase enzyme. So carbonic anhydrase enzyme, what we have, this is the question number 35, right? We have OH2, we have histidine, carbonic anhydrase, right? Histidine and then histidine. And what this is, it involves in peptide bond cleavage. Carbonic anhydrase won't involve in peptide bond cleavage. So option A is wrong. What about option B? Redox, inactive zinc 2 plus ion is involved in the catalytic activity of this enzyme. Okay, option B is correct. Activated MOH2 metal ion act as a nucleophile in the enzymes. Yes, it is acting. Okay, C, MO2. So option C is also correct. The metal ion is coordinated to the side chain of histidine residue. Yes, it is connected to histidine residue also. So option D is also correct. So likewise, if any other questions are there, you can directly ask me. Okay. Otherwise, I've solved this. This unit is very much easy, and it is a very small unit. Within 1 hour, you can practice it. So do practice. Do well in your examination. Thank you.